Okay, hello everybody. I'm Rob Burgess and I'm Chief Business Officer for Sinobiological, and I would like to officially welcome each and every one of you to the next installment in our series entitled Lock and Key Immunodetection Webinars. So with that, I want to just first briefly mention one housekeeping item here, and that is we will be accepting questions from the audience. If you would just simply type your question into the chat box. At the end of the seminar, I will take those questions and present them verbally to our speaker, who will then have a chance to answer them. So it is my honor and pleasure, without further ado, to introduce to you our honored speaker today. It is Dr. Chun-Nan Chen. Dr. Chen is the Chief Executive Officer of Single Cell Technology Incorporated. Dr. Chen is a research scientist and a business manager with over 20 years of experience. He has held senior scientific positions with Solera Genomics, Applied Biosystems, Pharmagenesis Systemics, DesignRx, and DesignRx Pharmaceuticals. He also taught at the Advanced Genome Sequence Analysis course at Cold Spring Harbor Laboratory. Dr. Chen received his postdoctoral training in biochemistry and molecular biology at UC San Francisco. He obtained his PhD from Michigan State University and his bachelor's in science from National Taiwan University. Dr. Chen is also a registered United States patent agent. Dr. Chen, the title of Dr. Chen's talk today is High Content Multiparametric Antibody Screening for Specificity and Function with AbTheneum. So Dr. Chen, I will turn it over to you and allow you to share your screen and begin your presentation.
Thank you, Rob. Stop sharing here. Okay, I want to continue. Yes. And here is the screen here. Can you see my screen? Okay, good. Thank everybody. I also want to extend my appreciation to Sinobiological for allowing me the opportunity to introduce our company, also our technology platform called AbTheneum. Let me just dive right into it. So we are located in the San Francisco Bay Area. We started pretty early. This is when the notion of single cell analysis is still not mainstream yet. Now, obviously it is. But so it took us a while to really get all of the technology to put together. We have some of the most sophisticated technology, including next generation sequencing and DNA microarray that has been two of the ancillary pillars of our technology. And we spend time develop and mature a proprietary screening antibody screening platform that is called AbTheneum. And AbTheneum is a play on the word Athenian. It's a Greek word that stands for an institute of great learning. And what we want to imply with the term AbTheneum is we want to learn using our platform a great deal about the antibody response from a host here. The platform itself has a really flexible host range. We have been able to work on mouse, human, and rat, and getting their antibodies from their responses. Also, we are currently working on getting antibodies from rabbits. We have been able to mature the platform in 2016, and we have been working for helping the global pharmas for their antibody campaign for the past 5 years. And we have quite a few repeat customers. And with a lot of these customers, they have already moved on to the transgenic mouse line, such as OMT, Triani, or Alloy. So we have been working on all those three major transgenic mouse lines. And we really specialize in some of the challenging project. And one of the, I think the example is finding epitope that is in the equivalent of a peptide in the multi subunit complex. So that's a very tiny epitope to hit. And we are able to use our technology's advantages to help the customer find antibodies against that tiny epitopes. And we have a number of patents, a total of 12, including 5 that are issued in the US and 2 in the EU. And they covered anywhere from the core methodologies to the associated instrument as well as the applications. And here is the workflow of AbTheneum. So it's broken down into 4 parts. The first part is having after the immunization and taking the hyperimmunized animals, taking their lymph nodes and making it into single cell suspensions. And we use a kit based on a CD138 markers to enrich for plasma cells. And these cells are the terminally differentiated B lymphocytes that will secrete antibodies natively at a high rate, up to 2,000 molecules per second. So they really crank out these antibodies. And we go clonal from the very beginning. So we deposit these cells into what we call the Picoliter device. Where's my mouse here? Yeah, here. So you can put them in. These are using limited dilution. So by Poisson distributions, most of the wells are empty. And then whenever a cell, whenever there's a wells that has a cells, that is typically a single cell. And so we have them in the culture for a short amount of time, and we put on a capture surface. There's something I'm going to illustrate in the next few slides to capture the antibody to generate a micro antibody array from that. It's like a printing process. And that antibody array will be analyzed with a fluorescent labeled antigen as well as some other screening molecules. And so that's the second part of it. And the third part is one of the most challenging portion for our technology development is to lyse all of the cells on the device, capture all of the cells, barcode them, and then sequence all of them using next generation sequencing. So that part, I'm going to go over that a little bit. And the fourth part, of course, is to combine the information we gather in the second part as well as the third part, so that we will collate that information and then help the customer as well as ourselves to pick out the antibodies of interest. And then to recap, we separate the cells to culture and we capture their antibodies into the arrays. So we can actually analyze them offline of this workflow here. So the rest of can go on and we can actually capture the mRNA from that without worrying about the well-being of these cells. And I think that's the biggest difference of our technology versus so many other competing single B cell approaches. So the ability to separate the antibody from the cells so that you can analyze them at will at any time in some other places and with using some harsh reagents when possible. And then the other thing that is different from all others is we don't pick out those hits representing the single cell that represent the hits. We sequence everything on the device we have, and then we match up the information in the back end. So this allows us a lot of flexibilities and also the high throughput as well as the ability to really mine the antibody response on the different animals. And all of these assays are done in parallel. That's why it's very streamlined and efficient. And we do all this in a matter of a month. And here is some of the workflow. I'm going to dissect it. So we have the cells deposited in the cells, and you can deposit in these microwells and then start to secrete antibodies. And you can see that it's still started to fill up with the antibodies. And then on the other hand, you have a functionalized glass slide that is able to capture the antibody. So you put it on top of it and you make a seal over these, basically a lid over these different wells on the picoliter device, and then you separate them. And then the antibodies are matching the footprint of these cells. And you can see if you look at it from the other side, it's a mirror image of the cell's locations. And what is interesting is these cells are still alive. They still continue to secrete antibodies. So you can actually make another one. And in fact, we have been able to make up to 4 slides. We haven't really pushed the envelope, but we're able to create 4 replica slides of these. So this allows us to screen the molecule of interest in any number of ways possible. For example, you can screen for cross-reactivity by looking at the staining pattern on one slide versus the other. For example, we can also search for other type of attributes such as specificity. And I'm going to go into some of the examples later. And then obviously, once you have that, then you can actually flow the fluorescein label either antigen or any sort of screening molecule. So that will basically bind to the antibody interest. And so you will identify that by scanning in the DNA, actually microarray scanner. And you can do that for some other slides that you have done. So that's the sort of the second part of the antibody capture that I talked about earlier. And this is the third part that is capturing the mRNA. And in fact, this is the paired heavy chain light chain paired mRNA from each one of the cells. We are not capturing the transcriptome. We are mainly targeting the heavy chain light chain from these individual plasma cells. And we have designed a custom DNA microarray. This is a crude representation of that. In fact, this only shows 25 of these features here, and each one of the feature has millions of these oligonucleotides that are specified by our bioinformatic groups. So each one of it has a unique barcode in it at each one of the locations. And then similar to the antibody capture ones, we actually capture the lysis cells and capture all of the mRNA from that. So we end up capturing the mRNA when we do cDNA synthesis directly on this chip, so thereby incorporating the barcode on each one of the features into the cDNA. So after that, we'll elute all of the cDNA from the microarray, amplify that, but add on the Illumina adapters, and then send it off to a sequence service provider to get the sequence information. So this is what we do. And obviously, after you do the sequencing, and this is again a crude representation of that, you actually construct a synthetic map of all of the sequences of the mRNA from that particular wells, so you can match up with the antibody scanning pattern that you have. So then in this case, this red mRNA one correspond to the pits that we have if you happen to use the antigen as the screening molecule to flow over the antibody capture array. So I hope that is clear to everybody. And then let me introduce you to some of the mode of screening in the antibody capture portion. So we call it simul screen because we do these antibody screen simultaneously here. So first of all is a ranking assay by normalizing the amount of the signal. If you have that on each one of the spots of the antibody capture, as well as flowing the antigen or the screening molecule at different concentration, you can have a rough ranking of the affinity of the antibodies that is captured on the slide. And the second one is, I talked about, is when you have multiple of these slides, you can interact with or interrogate it with different molecules, such as the human antigen that you use for immunization, as well as the monkey protein. So you can look for cross-reactivity that way. And the third one is specificity. You can actually look for something that you don't want initially. So if you have a single slide, you can react with something that you don't want. And then subsequent to that, if you see any of the new spots, those new spots potentially will represent molecules that you want. So this type of simple setup is one way that we can look for specificity. I will have a case studies in the back end. And the fourth one is really kind of exciting for us, which is developed last year during the pandemic here, is the blocking assay. So we can actually screen for blocking activity during this campaign. So that allows us to really quickly move from the hit to the lead for a given given attributes for those antibodies. So I will have another case study for that. And then the other part of it, it's the extension of that, is we start to add this. And then recently we demonstrated that we can, instead of capturing the antibody onto a slide, we actually ended up capturing using a monolayer of cells. So you can actually look for antibody that bind to the membrane protein that is present on the cell surface. And you can use engineered cells or native cells, depending on which one serve the purposes. And because these cells also ultimately going to be, move away from the cells that producing the antibody, the plasma cells, we're like moving toward doing a functional assay. So let's say the cell binding assays that you set up with a reporter cells of some sort, and you can actually look for signal transduction event that you have a particular readout to look for, say, agonistic or antagonistic or even allosteric activity of these antibodies. So that's something that we are planning to do toward the end of this year. So we have this multiple mode of screening this antibody for their attributes. And plus you can, on the other side, you can get the sequence here. So that allows us to really look at these antibody in a very broad way and very deep because of the high throughput nature of our platform. And here's a picture of our Picoliter device. It's a little bigger than a quarter here. And you can see that it's roughly divided into three portions. We call it the active area. And each one of them has 30,000 microwells that has the volume of a little less than 200 picoliters. So that's why we call it the Picoliter device. And here is after we print it out, and then to stain it with, first of all, the green is actually a antigen that has been fluorescent label with side 3. And after scanning it, it shows up in green channel. And then the second one is actually a separate image after we stain the universe of these antibodies using the secondary antibodies, gold anti-mouse IgG, for example. Then you stain all of the antibodies producing cells, their footprint on everything here. So you're able to tell the specificity here that it doesn't really bind to all the other spots. It only binds to these specific spots here. So this is how we do. And in terms of the capacity, we will screen up to 25,000 cells that could get sort of interrogated on a particular run here. So we're able to look at these multiple of these binding profile with phenotypic characterization, as I mentioned before, using SimoScreen or SURF screen. So this is when you sequence all of these cells, and this is again a representation of what it might look like. We sequence all of the cells here, and what I'm representing here is using the heavy chain CDR3 on the top, and then the light chain CDR3s at the bottom to imply that we actually got the sequence of this particular spot. And then our recovery rate right now is about 70% to up to almost 90% of the cells that once we get the IgG spots, we'll be able to recover the paired mRNA sequence for both the heavy chain and light chain. So we get the full length of the entire variable domain. So you're able to take that sequence and reconstruct them and to do your downstream analysis. And so once you filter away all of the sequences that don't match the stained spots, then what you end up with is the antigen specific antibody sequences here. So you can see that a particular family started to emerge, that we call it 1 here. And this is family number 11, if you will. And so you can see the power of sequencing everything and just combining the information of the antibody binding profiles as well as the phenotypic characterization with the sequence information. So that allows you to look at the entire repertoire of the response. And so that's the power of such a, I would say, printing and linking, or the registration of these matching allow us to do. So that's the sort of the gist of our technology on the sort of the broad way. And here is a operational advantages, that because everything is done in parallel and it's in the micro scale here, you really don't need a big team to do that. And in fact, we're able to do a particular run is doing the harvest on a Tuesday, for example. Where is my mouse here? So after immunization, this is typically done on a Tuesday. So the enrichment is taking the lymph nodes, the lymphocytes from lymph nodes, and isolate the plasma cell population from that, and we spread that onto our Picoliter device, and we do our antibody capture, and depending on how many slides you want to capture, so it can be a little longer day. And after that, you lyse cells and capture the mRNA overnight for convenience. And the rest of the week, we will split it into 2 workflows. So on the one hand, you'll be interrogating with the fluorescently labeled antigen with some other screening molecules. On the other hand, we'll be doing cDNA synthesis in situ directly on the capture microarray, and then to elute that. So once you do that, then the cDNA is stable. You can store it and to look at the antigen interrogation results. So let's say for whatever reason, the immunization did not go too well, you only have very few hits, then you don't have to go forward with this particular workflow. But let's say you got quite a number of hits and the binding profile looks very exciting, then you can move forward with the cDNA sequencing part by doing the NGS preparation. So there's a stopping point at this point, and you can decide if you want to go forward or not. And the rest of it takes another 3 weeks or so. And so within a month, we actually get the data from since the harvest, the lymph node cells from the immunized animals. So at that point, we will work with the partners to decide which one to reconstruct, and we send it out for reconstruction and recombinant expression for the antibody of interest, so they can be looked at. So in this particular example, I think this is from a different slide, you can do an ADCC assay with these reconstructed recombinant antibodies. So you can look at the landscape and lineages of these antibodies, and you can validate, as I said here, the least number of clones with the maximum diversity, since we know the sequence of all these. The other thing that is also not to sort of just put another point is, since you have the sequence and with a little bit of characterization, you can immediately file patents. If this is a fast race, then at least you can file patents on the compositional matters for this antibody sequence. So that's another advantage of having the sequences up front and to be able to do it in part of the analysis. I think next ones are the studies. Let me see my time. Okay, I think I'm okay here. So here is the one part of the data output that we have. So this is an example of the BCMA that we have run before. I want to use this as an example. And so you do a cluster analysis. So what does that mean? So let me bring up this. Is what you do is you combine the heavy chain sequence and the light chain together into a text string. And then you ask the question, how is this different from the next sequence that you have? So in this particular example, if you treat the first one on the top as a reference sequence, then you can see that, oh, the second one is off by one amino acid here. So then the added distance of that is considered one. And so you can link it up this way. And then if you look at that reference sequence as opposed to the third one down here, then you can see there are three different ones as different. So different from the first sequences. So the added distance would be designated as 3. So obviously I have this in the horizontal sort of the configuration. So here, because of the space constraint, I put it on the vertical ones. And you can see that I actually put the antibody in different color to separate them if it's a particular family. So a lot of them are a singleton. We call the singletons here. So let me put out another one. So here in this particular campaign, I think we recover 300 some clones with paired sequences, and it comes down to 191 of the unique sequences. So when I say unique, is some of them could be off by just one amino acid, but they're still unique. But if you cluster them and looking at the relatedness, then there were the total of 79 of these families. And those actually will include what I call the singletons. So what do I mean by singletons, and how do we actually put the designation is that we put something like a 0 here because they're not related to anybody else. And here is it's a small family of 2 here. And so depending on how they showed up here, this could be family number 1. And this is a bigger family here. So this is just designated number 6 here. So these are another few of the singletons I have. So it's designated as 0 here. And this is another family called designated 7 here. So I just want to show you so that the next few slides will make sense here. So this is the type of analysis we do. And so let's go into a case study. So here is a studies where we want to find antibodies bind to a multimer, not the monomer. So this particular protein has, the subunits here, but then the goal of the project is to find antibody that binds to most likely the interface between the different monomers. So it just wanted to bind to the multimer configuration, not the monomer. So obviously this is something that is quite challenging because most of the time you're gonna, even though you inject the multimer into the animals, it's gonna find antibody that binds to the monomer because that is part of the composition here. So how do you find that? So we do that with the different slide I talked about. So if you have 2 slides here, so you can interrogate one of the slides with the monomer, the other one with the multimer here. Then you can actually match them together and find which one of the slide has the multimer that is specific to, a spot that is specific to the multimer, not the monomer. So that's one way to find multimer specific antibodies. And obviously you want to have the full length paired heavy chain light chain sequences to that. So this is at least deceptively simple case study here, but to find these using some of the conventional technology that is very labor intensive, but we do that in a parallel fashion. So we do it in a month. So here is the number of antibody we found. And let me just guide you through this. So here is the cluster ID I mentioned. So remember the zeros. That means that these antibodies, they don't look anything else of the other antibodies here. And you have these numbers here. So apparently they have up to 93 different families here, so that are related sequences. And so anyway, I have a call coming in. Let me silence that, otherwise I get distracted here. And so here is the binding profile I said. So these are the one that binds to the monomer, but not the multimer. So there's a lot of that here. right? Obviously. So here's the corresponding amino sequence for the heavy chain VH and VL. So this is a relatively simple studies here, and this is the ID that just the index of different clones up to 886 paired sequences that we found from this particular campaign. And if I do some sort of filtering of that, is if I filter the one that looks for just the ones that bind to the monomer, I find 868 of the records out of the 868 out of 886. So obviously most of the antibody that you find will be against the monomer, which is not too surprising because that's the basic unit of it. Even though you injected the multimer into it, they will bind to epitope that is present on both the multimer and the monomer. So, but if you go in and do the filtering and look for the ones that are binds to the multimer, but not the monomer in this particular case, then you end up with 13 of it. Obviously, it's going to be rare because most of it will be binding to the monomer. So there, we find 13 records of it. So if you look at this more carefully, so there are, 6 of them belong to the cluster 0. So that means that these antibodies are unique. They don't look they don't look like anybody else. So they are actually in this sense is a family. It's a single member family here. So there are 6 different types here, and then 2 is another family here. 2 from that particular family was found, and maybe most likely the family only has 2, because if the sequences are very similar, they probably have very similar binding profile. And but obviously, if you look at this 14, where's my mouse? This obviously came from a family. So for interesting reason that because of the sequence differences of these antibody family here, this one member 14 in the family ID 14, this particular antibody will bind to the monomer, but not to the monomer. Bind to the multimer, but not to the monomer. But there are obviously some other members will bind to the monomer here. So, and you also look at the 90, which is another family here that binds. So if you look at the diversity, there's 6, 7, 8, 9, 10. The 10 different types of antibodies that you want to basically work on them further. So at this point, because of the sequence information, you already know the diversity of the antibody you're looking at. So if you're happy with this diversity, then you're done. You can move on to it. If you're not happy, you can do another run, try to increase your diversity of interest. So this is I think one of the biggest benefits of having the antibody sequence up front. And so that's the first case study. The second one is obviously for us to look for the COVID-19 virus, the spike protein of it. And so what we want to find is antibody that will block, for example, interaction between the human ACE2 and then the receptor binding domain of the spike protein on the coronavirus, the novel coronavirus. And so here is what we do. The campaign is doing the blocking assay. And so we put the cells in the wells, like I said before, and you secrete antibodies, and you come down with the glass slide, you capture the antibody, just like I said. But you can also capture another slide here. And you do the second slide. And then you go on and interrogate them slightly differently, as we're going to see. So you're going to, you interrogate with the unlabeled receptor binding domain. The other one is labeled. So you see some of them binds. And obviously they have to bind to the same spots because that's the one that are antigen specific. And then you come on and using the label human ACE2 protein. And then some of them will bind to it. And you can see that the one that binds to the RBD obviously is the antigen specific antibody. And then the middle one are the non-antigen specific ones. And you could see that this actually will be the non-blocker because it allows the binding of the ACE2 even upon binding of the antibody to the RBD. And the other side is the blocker because once it binds to the RBD, it doesn't allow binding of the ACE2. So this is part of a blocking assay setup here. So what I do here is I'm gonna take out some of the cutouts, image cutouts of it. And so for the non-blocker, so this is from the slide 1, this is slide 2. And you can see clearly that it has signal of both of them. These are the ones that we are not interested because most likely they are not going to be neutralizing, for example. And this is the one that you can see that the slide 1 is have very little signal, if any, but slide 2 has strong signal. So those are the ones that we're able to quickly find if you use the blocking as a functional sort of surrogate here. Then these are the part of a functional assay, if you will, to look for it. So we're able to do this assay very quickly on the antibody that we generated from a cohort of mice and then move on with their analysis. And we're able to find over 1,700. These are antigen-specific antibodies, but because of the space constraint, I was only able to show in this particular slide 539 of them. Okay, and then so these are all antigen-specific, and you can see the edit distance as I explained before. There's a 10 and 20. And most of these belongs to a family. So they have, someone that has a really large family here. So, and then we reconstructed. So the one that you may or may not be able to see a magenta line here. So those are the one we selected for reconstruction. So we reconstructed something like 17 of them, and every single one of them blocked, show neutralization activity when assayed later. Oh, this one we don't have to explain. So this is the type of assay we did using the pseudo virus that at the time incorporated the D614G mutation in. So this is the one of the earliest variant that shows up. And it's pretty simple. Oops. Is if you have if you bind with the antibodies and it doesn't allow to infect, then you don't see signal because the luciferase mRNA did not go in there, get translated. And if it does allow infection, then you actually show signal. So this is the type of neutralization assay you can do in the BSL-2 labs here. So we did that, and we took 7 of the best of the 17, and we're able to do 2 different kind of assays. So one is doing the live virus assay. This is still with the Wuhan strain because the variant wasn't available. So you can see that and we used some of the other FDA approved antibodies, the benchmark. So these 2 are from the Regeneron antibodies. So they're in broken lines, and this one from Eli Lilly. And you can see the antibody that we generated, they're on par or sometimes better than the Regeneron antibodies. But when you test them against the pseudovirus that is representative of the South African variants, then you can see the differences here. In fact, the Eli Lilly ones stop, neutralizing the virus as well as one of us, but we have at least 2 of them that remain strong and actually better than the Regeneron here. So this is a type of assay that we're able to quickly find using our blocking assays. And so in summary, so AbTheneum allows us to analyze these single cells in the massively parallel matter fashion. So it gets really a lot of information about each antibodies before we even sequence that. And we sequence all of the cells. There's no picking out there. So we don't have to do anything in real time. We get a lot of information about their binding profile, and sometimes a really difficult one like the multimer specific but not monomer. And then the blocking assay that I just explained allows us to quickly go from hits to lead. And in this case, the neutralizing antibodies that saves times and then also increase efficiency. And I'll stop here and then I will take questions.
Thank you, Dr. Chen. That was an excellent seminar. We really appreciate you putting that together for us today. And we've got a good, strong turnout today from the attendees and some excellent questions. Since we're limited on time, we'll try to get to as many as possible, and I'll just run down the chat box and start with those who asked first.
Okay.
The first question is from Don Gwent. And it says in step 1 of the workflow, how long are plasma cells kept in culture before step 2?
Very good question. So that is, we only have it, the isolation itself takes an hour and a half. And once we isolate these cells, we don't really get them to recover. Basically in the middle of 5 minutes, then we already put them in the Picoliter device and start capturing. And the capturing time ranges from 40 minutes to 50 minutes, and sometimes maybe in an hour. So depending on how many of these you want to capture, and then obviously the more you do, we found that they already secreting antibodies. So the subsequent capture could be less time. So the overall time that after we get the single cell to we lysis cells is no more than 4 hours, I would say.
Got it. But the cells need to remain alive in order for the RNA to remain intact, right?
Yes, yes. So I say 4.5 hours. We
Right.
kept them in the device before we lysed them.
Okay. Sounds great. Next question is from Colby Salters. Colby asks, how often do you see multiple heavy or light chains from a well with a single cell?
So right now we actually have a what is it called? The automated microscope. So we can actually pick out which wells that has multiple cells in it. And then I would say typically it's if you do the type of the limited dilution we have, 85 to 90% of the wells when we find them have single cells in it. So the rest of it will have doublets or triplets there. But those are the ones that we actually, first of all, we identify them by the microscope images. And second of all, because of our sequence recovery rate, if we recover anything from that, we will recover sets of 2. If there's double cells in it, we'll recover sets of 2 heavy chain and 2 light chain here. So that one may say that's not very useful, but sometimes what happens is you will look at all of the sequences here. If you see sequences on the other side is a singlet, a single cell that has related sequence, then you probably will be able to deconvolve some of the wells that have double cells in it. So we actually frequently do that. And of course, if it's not antigen specific, we don't care. A lot of times they are not. So when it's antigen specific, then we actually will look at the related sequences and we actually don't throw them away. We might be able to deconvolve it, and actually sometimes we're able to reconstruct them and get successful clones of it.
Great. So it sounds like cross-contamination between wells is not a major concern, but if it happens, you have methods to deal with it, right?
That's correct.
Okay, great. And Don Gwen has another question. Can this approach be applied for transcriptome in addition to repertoire?
That's interesting. We obviously we haven't had that request from the customer, and all of our customers they're interested in getting antibody sequences. So we haven't tried that. I think that will be interesting, but on the other hand, you want to know what you're going to be looking at, right? So and here, if you're looking at transcriptome, you want to be able to associate that transcriptome with something else, right? If you just want to look at transcriptome, then I'll say 10x genomics approach is that you put the cells into droplets and sequencing. That will be enough because you're not associating that transcriptome with something else. But I noticed that right now with some spatial genomics, they actually try to correlate some position of the cells in the tissue, for example. So then that makes sense. Then if you already have some other information here. So when we have application where the whatever cells does, for example, it secretes certain cytokines, and maybe you want to correlate that cytokine secretion type with the transcriptome, then I think that that could be an application, but we haven't done it.
Right, right. But in general, for transcriptome analysis, there are other platforms that may be more adequate for that, like 10x Genomics' platform, right?
Yes.
Okay, great. And Moen Shayagania asks, does increasing your specificity come at a cost of reducing your sensitivity?
Increasing specificity. I don't quite get that questions here. So the when we look at the specificity, we obviously will interrogate the antibody at different concentration of the antigen. And if we don't see a signal here, obviously we're going to lose it. So I think the specificity is really an overall limitation of the fluorescent label antigen, the molecule we have, and also our detection mechanism, like the DNA, the microarray scanner. But as far as I know, the microarray scanner is one of the most sensitive instruments available. So it's basically a confocal microscope with a very little distance between the lens and then the surface that emits the photons. So I think it's one of the most sensitive device here. So I don't think there's any sacrifice being made here. It's strictly the limitation of our system or the instrument that is used for detection.
Right.
I don't know if I adequately answered that question, but that's my answer.
Kind of a tough question. Sensitivity versus specificity is always tricky, right?
Right.
Um, so Don Gwynn has another really good question. Her third question is, can this capture cytokines that plasma cells release?
Yes, we actually looked at T cell. We haven't gone full force looking at that, but we do have a longer term project to look at cytokine secretion profile, because if you want to characterize different T cells, you want to look at the different subset of it. And so the ideas would be looking at what they have, and with the microscope we have, we can also look at what's being expressed on the surface. So this is again not what we're doing right now, but the longer term we want to look at T cells for their secretion pattern of the cytokines, as well as what kind of markers is on the surface, and also their paired alpha beta chain for the TCRs. So that's something that we've been thinking about doing, but we haven't done it yet.
Great. And Ching asks, and maybe you had already presented some examples of this in your talk, how do you do functional assays with antibodies captured on the slide?
You don't. So all of the functional assay we have is, instead of, let me go back here. That's a good question.
Glad she asked.
Yeah, so here, if you capture the antibody on a slide using whatever reagent, that's kind of hard to do that. But imagine instead of using a capture agent to capture the antibody here, imagine that this slide is covered with cells, right? So you're looking for the antibody that when it binds to the cells have some sort of readouts, some fluorescent that goes and trigger the reporter gene inside the reporter cells. And that's the kind of functional assay that we'll be looking at. So the capture sort of setup would be totally different. So this is more related to the Simul screen here. So we have to remember that like this, we can use one slide to capture antibody onto the slide and interrogate it just like we do with the different fluorescent label molecules. But the second slide could be a cell-laced ones that will capture the antibody onto the surface of the cells, and we look at the readout of the cells here. So we can do the surf screen and the Simul screen at the same time. So there's no exclusive nature of that. So that's how we're going to be doing the functional assay.
Great. And George Ivan Cervantes asks, can we use this kind of screen in a phage display?
Phage display. Well, the issue, let me go back to the screen here. So we get the cell directly from the hyperimmunized animals. So we don't do any sorts of other manipulations. So we don't do fusion. So it's not hybridoma. We're just directly taking the cells. They're already secreting antibodies. So at that point, it's easy enough to capture the antibodies. So if you're talking about phage display, that means that you already have done something in the front end. They mix up the heavy chain light chain. So I think the key philosophy of our approach is to um, capture the native heavy chain light chain pairing, so that when you go through this, every cell is confined into a particular wells and we capture them. And obviously what you get back of the pairing is their native pair. And then so far, this is why people been using hybridoma, because despite the difference in the approach is still you're capturing the native of the pairs. And phage display is just a different philosophy. You don't really care about the original pair. You randomly pair them and you're hoping that you're going to find that. So in a sense, if you have it in, for example, phage in a, um, I presume you can actually put, instead of the cells here, you can put the individual phage there. But I guess the bacteria that deals with the phage, because we want to be able to capture the antibody from that. So I think it will be a little clunky. So I wouldn't recommend it. Yeah, I think you can make it work, but I think the fact that you already gone through the process of mixing up the heavy chain light chain, so I think you might want to stay with whatever that works for the phage display.
Yeah, sounds like phage display is just not necessary.
Right.
Colby Salters asked, what percent of the plasma cells end up secreting IgG?
Oh, that's a very good question. Um, it depends. Right now, with the CD138 cells, CD138 markers, the percentage varies a lot. And we don't know if it's simply the strain that we use, the adjuvant, or the immunization conditions. So it varies from sometimes 10% up to 80% of the cells that we put on. So even though we put down, 25,000 cells, not all of the cells will be secreting that. So this is a really good question. And we right now don't have a good handle on that. But we recently acquired a flow cytometer, the cell sorter. And so we're looking into some other type of mostly non-lineage markers to be allow us to select for cells that are secreting antibodies. So regardless of whatever CD138, we want to just find cells that are secreting antibodies. So we have some ways of doing that. So if when we get that done, I think then whatever number of cells that we obtain, a vast majority of it will be secreting antibodies. So right now, I think we're constrained by the marker that we use for selecting plasma cells.
Right. And Moen Shayagania has the question, how repeatable is the binding profile?
It's very repeatable because we actually have done multiple of that slides going back here. Here. So you can actually, we have done that with the same antigen. If you do the staining on both of them, in fact, on even 3 of them, we see completely identical patterns. So this is at least one demonstration of the, uh, the, the consistency of the binding assay that we have. And also, and the other side is we also get the sequence, and we also will be able to match the sequence to all of the binding sites. And sometimes we'll see that, wait a minute, this particular sequence is present on both of them, but only one of it is positive. Then we actually look very closely at that particular spot. And what happened is sometimes it's very, very weak. And then when you go through your image analysis, sometimes you miss that. Or sometimes there's absolutely no signals. And what we found out that it's the sequence differences. Let's say there's a minor differences in the CDR3 that causes the sometimes complete loss of the binding activity. So we actually see that, and that's the benefits of having the binding profile as well as the sequence at the same time. And you can go back and re-examine what you have done in terms of the binding assay you have.
Great. And here's a question from Yongjian Wu. I think this is related to the efficiency of percentage of IgG secretion, but on slide 20, you mentioned good versus not good immunization, depending on how many hits you get. So with a good immunization, what's the percent positive of the total wells that show antibody sequence?
So I will say the good one, I will talk about the immunization efficiency. That means whatever number of IgG that we have. So as a function of as a percentage of those IgG secreting one, we have as high as 27% of the IgG secreting ones being antigen specific. So those are the highest one we have seen. And most of the other ones ranges from the low ones we have seen is 2%. But then I think most of them hover around 10%, 9 to I say 12% of them will be antigen specific. So if we spread, so that means that if we spread, 10,000, if able to see 10,000 IgG secreting one, then the 10% will give us roughly 1,000 of the antigen specific spots here. And if we recover 70% of those sequences, then we get 700 sequences. And it's more than enough for any of the group to looking at that. In fact, they always sometimes complain we have too many sequences. So this is why the binding assay is very important. So you want to do something like the multimer versus monomer type of assays because you really want to look for something that is specific. If you're strictly looking for a binder, then I'll say any of the other traditional approaches would be fine. But if you want to look for antibody with very specific binding profiles, or you want to quickly find the one that has some function like the blocking activity here, then I see our approach obviously have a pretty good advantage.
Yeah, yeah, great. And I guess having too many sequences is a good problem to have, right?
Sure, yeah.
And you also target multimers because I guess they look a little more in vivo like what occurs in nature. And so it's something that would be more biologically useful in the long run, right?
Yes, yes. And some people, they really look for special epitopes, the one that are difficult to target, but is biologically or therapeutically relevant. And they somehow, so this is where we're not the expert of it, but for those people who are really into target biology, they've really figured out that that's the epitope I want to target. And so those are the type of more challenging project that we ended up getting.
Right. We're just about out of time. Just a couple more questions. Well, these are from Ruifa, and I think you may have addressed this in your talk, but the first one is, how do you sort single cells into the picoliter device?
We don't sort. We just we do limited dilution. So it's a Poisson distribution. We control the cell density, and they just fall down to the wells by gravity. So that we don't do anything special at this point. Yeah.
Simple approach, but it works, right?
Yes. It takes no longer than 5 minutes. So you really want to move fast because the plasma cells, they don't stay around forever.
Right. And then the second question, we'll make this the last question because we're just about out of time, is what is the principle of binding antibody to the glass slide and what is the binding efficiency?
We use different type of agents. A lot of times we use just off the shelf secondary antibody that works fine, but sometimes the project demands that they use some sort of other binding agents, sometimes proprietary. So for what I'm seeing, the secretion rate is so high that the inside the wells, within 10 minutes, based on our calculation, it reaches 10 nanomoles or more. So most of these binding agents that we have is pretty good, especially some of the secondary antibodies from goat or some of the other animals. Those have an average KD of 1 nanomolar. So the binding sort of the capacity and efficiency is quite high.
Great. Well, thank you, Dr. Chen. I specifically want to thank you for an excellent seminar today and for an even better Q&A session. I feel like we had some great questions and we appreciate you taking the time to answer those in detail and more importantly, joining us today and putting this together in sharing this exciting technology and sharing a little bit about single cell technology incorporated as well. In addition,
Thank you for having me. Yeah.
oh, thank you. In addition, I want to thank the attendees. We had a very good turnout today. We appreciate you all staying for the entire lecture and for the wonderful questions that you posed. I want to mention that in just a few hours, there will be a recording of this webinar linked on the Sinobiological website, so you can see it again there if you choose to do so. And I also finally want to thank my colleague, Dr. Jen Liu, who's our marketing manager, and she organized and helped to host this seminar today for Sinobiological. And so with that, I will officially close the webinar. Thank you again, Dr. Chen, and everyone have a wonderful rest of your day or evening.
Thank you.